So Belantamab, Carfilzomib, Lenalidomide, and Dexamethasone is an investigator-initiated clinical trial that we performed at the Levine Cancer Institute. We enrolled patients after at least one prior line of treatment. Patients received Belantamab mafodotin at two doses, 1.4 milligrams per kilogram and 1.9 milligrams per kilogram in a phase 1 dose escalation with dose expansion included in the trial, in addition to KRD standard doses as approved for multiple myeloma. In this meeting, we report the final results of the phase 1 portion of the trial. We showed that the regimen was very well tolerated. We were successfully able to give Belantamab mafodotin at 1.9 milligrams per kilogram every other cycle. We did not see any dose-limiting toxicity in the first initial 6 patients, as well as in the expansion cohort. Safety signals were as expected, mostly hematological, in addition to keratopathy, which was reported in about 95% of the patients. All keratopathy incidents were reversible, and it was manageable by either dose reduction or dose delay. Mostly we used dose delays for our patients. And I would say 100% of the patients received the first dose. For subsequent doses, it ranged between 30% to 70% of the patients were able to receive their subsequent doses of Belantamab mafodotin, with the majority getting it more than 50% of the patients in subsequent dosing. In regard of the efficacy, Belantamab mafodotin showed really good efficacy results when compared with DREAM7 and DREAM8 trials, and that kind of encouraged us to proceed with this protocol, especially for the future for high-risk patients. During our phase 1 results, we had 100% response rates. Close to 60% of the patients were stringent complete response. Out of those, also, we had more than half, about 60% of the patients were MRD negative 10 to minus 5, and a significant portion was also MRD negative 10 to minus 6. Suffice to say that the regimen was effective in Len refractory population, as well as showed good efficacy in patients with high-risk cytogenetics, which was present in about a third of the patients, roughly. Given those results, we are proceeding with this regimen for phase 2 portion. In phase 2, it will be a multi-center study. We will enroll patients with high risk. Hopefully, will include the new definition that is about to be published from the IMWG for high risk patients. So our results will be consistent with the new definition of high risk disease, and we're very optimistic. Hopefully, we'll see good results from it. The regimen will include 4 cycles induction stem cell transplant for those who are eligible for stem cell transplant, and another 4 doses of consolidation, then followed up by triple maintenance regimen.